Using a separating funnel to separate components of a mixture is possible when the mixture has two immiscible liquids. Two immiscible means the liquids do not they are not soluble into each other and you can easily see their distinct layers. Now, principle used is immiscible liquids separate out in layers depending on their densities. The picture on the slide is of a separating funnel. We would also be doing this in the laboratory once you come back to school. So this separating funnel, if I add a mixture of kerosene and water to it, both having different densities, and I allow it to stand for a while, we can very distinctly see a layer of kerosene above the layer of water. And then we can remove the bottom layer using the tab that is given at the bottom and thus separate the components of a mixture where both the substances are immiscible. Where is this method used? This is used to separate oil from water, and also in a large scale it is used to extract iron from its ore. Think, is this method same as decantation method? I hope you remember what is decantation method. If not, you will have to go back and revisit certain concepts studied in the earlier classes. Sublimation is another special technique for separation. When is it used? When one of the component of a mixture is undergoes sublimation. Sublimation means that substance will directly go from solid state to gaseous state when we apply heat. Okay. Now this substance is removed. The mixture contains sublimable volatile component and non-volatile or non-sublimable impurity. Okay. We would also be covering this same experiment in the laboratory. Let us follow the flow chart which tells us what to do. Take a mixture of sand and ammonium chloride in a china dish. Cover it with a funnel that is plugged with cotton at its tip. Heat the china dish gently. We see vapors of ammonium chloride getting deposited on the inner surface of the funnel, which can be then scraped out. That ammonium chloride can be scraped out. So what remains behind would be a sand, which is a residue, which ideally is an impurity. Okay. Thus we can purify ammonium chloride by using sublimation technique. Think, do you know about a substance that is found at home which can undergo sublimation? I'm sure all of you have it at home right now. The hyperlink given below will also help you visualize how sublimation takes place. Chromatography is another method to separate certain components, to separate solutes from their solvent. The condition over here is the solutes are soluble in the solvent. So you cannot deploy other methods to separate them since they are soluble in it. Chromatography gets its word from a Greek word called as chroma. Chroma means color. So this actually tries techniques was used to separate various colors that are present in your ink. So the normal black ink or the blue ink that we use, do you think it has only black and blue component? No. There are various other colorful pigments that are there in that ink, which can be separated, which can be spotted using chromatography. How does that chromatography work? You can try doing it at home also. Take a thin strap of filter paper, draw a line little away from the base and mark a point at the center with ink. That ink, camelin ink, blue, black, it's fine. Make sure that that point is small enough and it's not huge. Otherwise, you will not be able to see distinct color spots. Place the filter paper in a glass jar or a glass tumbler having little water and ensure that the water level is below the spot or the ink mark. allow it to stand for some time as it is. Afterwards, you will see that the water rises on the filter paper because of capillary action, and it's as it moves across the filter paper, different colors are being spaced out or spotted on the filter paper. Some colors move longer distance while the others don't. What we can conclude from it is that the ink that we use, it's a dye, and it is a mixture which is made up of two or more colors into it. The color component that is more soluble in water will move faster than the one that is less soluble in water. Where do you think this technique is used? It is used to identify color pigments in a dye. Like we said, what is that blue ink made up of? Which all colors make that blue ink? It is used to detect drug in blood. You can also obtain pigments from natural colors using it. And in the forensic lab, that's a very, very good tool to identify the components that are present in a given mixture. The link below will help you to visualize how chromatography takes place.